This is the BioCentury Show.
Brought to you by the 12th China Healthcare Summit, returning to Shanghai October 22nd to 24th. Meet top decision makers and investors and get a first-hand look at China's dynamic life sciences ecosystem. Learn more at biocenturychinasummit.com.
Hello, welcome to the BioCentury Show. I'm Selina Koch, an executive editor here at BioCentury, and I'm delighted to be joined today by Nello Mainolfi, the CEO of Kymera Therapeutics. I've been looking forward to this discussion because Kymera is among the companies pioneering a new therapeutic modality, which is one of the most closely watched modalities, I think, in all of drug development, and that is targeted protein degradation, for which Kymera reported some promising phase 1 data in June. Nello is a medicinal chemist by training. He spent over 7 years at Novartis in discovery before he founded Kymera in 2016. So one of the promises of targeted protein degradation, I think one of the things that makes it very exciting, is the prospect of drugging the undruggable, right? And Kymera's lead program exemplifies that because it targets a transcription factor, a type of protein notoriously hard to drug with a conventional small molecule. Yet, degraders are themselves a type of small molecule. So I thought maybe just to set the stage, Nello, you could start with a description of the properties of degraders that make them able to, tackle these difficult targets.
So first, thank you, Selina, for having me here. I'm a big fan of the show, so it's always good to be also an active participant of the show. So starting with protein degraders, what problem we're trying to solve, and then how we can solve this problem. So as you said already, we've sequenced our genome more than 20 years ago now, and so we understand a lot about gene and also understand about the relationship between genes, proteins, and proteins to disease. And while we have amassed a huge amount of knowledge and information and data, we're still dragging about 15 to 20% of our genome and proteome. Regardless of the number of new modalities that have come to the market, going from obviously traditional small molecules to antibodies to oligo-based therapeutics to gene editing, we're still dragging generally at the same percentage or a marginally improved percentage of the proteome. And that's mostly because we're missing the right technology to go after targets that are in the cell, that are broadly expressed in the human body, that can be targeted in a disease agnostic, I would almost say technology agnostic manner. So what protein degradation brings to the table is an opportunity to go in the cell, in any tissue. So unlike, let's say, traditional RNAi that are limited to the liver, we can go to any tissue in the body and really present a disease-causing protein to the proteasome for disposal. Actually, the small molecules that we use for enabling degradations are actually not active. They don't have active inherent pharmacology. We're just presenting through a binding event the protein to the E3 ligase and the proteasome. So while small molecules need to find a binding pocket that is functional and it's eliciting a response, here we're only looking for a small molecule binding region that is, I like to say, inert, neutral, inactive, and through a protein-protein interaction drive a protein to degradation. So what we found is that traditionally undrugged proteins that are not evolutionarily designed to be blocked by secondary metabolites or traditional small molecules are perfectly suited to be drugged by a protein degrader because you can actually bind to many transcription factors, but it's extremely difficult to inhibit the function of a transcription factor. And so with a protein degrader, we can do so. And so transcription factor, multifunctional proteins, and others are best suited for a protein degrader approach.
And while we're on the properties of degraders question, I mean, I think an important one is you could kind of think of them as being catalytic, right? Do you want to discuss that?
Yeah, so our main effort in the past few years, and I'm only saying this, I'm sure eventually we'll get to it, or at least I hope so. But I'll mention it because I think it fits this particular question. So we've been working in immunology with the idea, with the thesis that with a catalytic degrader, we can block these pathways as effectively as a saturating dose of an injectable biologics. And so we can bring forth the concept of oral drugs with biologics-like activity. So how we can do that? So traditional small molecules, as you dose them, they have a Cmax in which you usually see maximal inhibition of the target. And then as the molecule is cleared from the body, you see less and less activity on the target. In a way, that's also true for biologics, that once you saturate the target with the injectable biologics, you get full inhibition. But because of these half-lives are very, very long, you get to inhibit that particular target with the biologics for a very long time. What we don't have a solution for is a molecule that engages the target only for a short amount of time, but is able to have its maximal effect. And so these degraders, as I mentioned, bind the protein and part of the UPS system, allows for ubiquitination and degradation. The same molecule can do that again and again and again. So that's where the catalytic nature comes in. We can degrade thousands of copies of protein with a single molecule. So what we need then, it's for a short amount of time, a relatively small amount of our degrader to allow the degradation to be completely degrading the protein in the human body for as long as the body resynthesizes the protein. What we found in most programs that we work on, that the resynthesis actually usually takes days and not hours. So we can give our drug once a day orally and degrade the protein completely for as long as we dose once a day.
Okay, before we get to your program, let's talk about the field a little bit more broadly, because so you started Kymera almost 10 years ago. And since then, there's been a lot of work on kind of two major classes, right? The heterobifunctional molecule, the PROTAC-like molecule, and then the molecular glue. By the way, heterobifunctional, that's a mouthful of technical jargon. I think you all should rename that. But in all seriousness, like they have different strengths and weaknesses, right? Heterobifunctional degraders, they're like modular in their construction, so you can rationally design them, right? But they break Lipinski's rules, right? They're large, they can be polar, they can be floppy, whereas molecular glues are these, small drug-like molecules, but I think historically they've been discovered serendipitously. So today, how do you think about those two trade-offs today? Is one like solving the challenges of one versus the other? Like, what do you think?
Yeah. So I think everything you've said is true for, let's say, the observer of protein degrader space. I think if you are an expert in the space, I think most, if not all, the challenges of making these heterobifunctional degraders into pharmaceutical agents that have the physical chemical properties, the stability, the half-life that will allow them to be dosed, once a day as an oral drug. Those have been solved by companies that have been in the space and have dedicated effort. So while at the really high level, again, what you said is true, that these are larger and they go beyond the so-called rule of five. We have figured out what the rules are, and we can make, as you've seen from our data in multiple programs, these molecules behave like active pharmaceutical agent with good properties. So the way we think about heterobifunctional versus molecular glue, I think about problems we're solving. So if we have a protein target that we have confidence in, and we think about highly validated pathway, undrugged targets in areas where we can show superiority quickly, when we have confidence in that, then the question we ask is that protein ligandable in a specific manner? Can we find a surface on the protein that can be bound with the small molecule? If the answer is yes, we will always go after with a heterobifunctional degrader approach because we believe it's the most rational drug design where you have absolute control of specificity, potency, and molecular properties. When the answer is no, this is a highly disordered protein, or the binding site that you identified is shared by other protein in the same class, then we resort to a molecular glue approach where we are trying to identify now a protein-protein interaction enabler that is a smaller molecule. So we like to think about from a technology solving the problem and not from can we get these molecules to behave the right way. We think we can do that regardless of the technology.
That's super interesting. Yeah. So you think the principles for making these drug-like are mostly been worked out. Yet when I think of the investment in recent years, it seemed like more of it's gone into the glue area. Well, what do you think explains that? Is it more just about IP or the types of opportunities, like you were saying?
I think it's the realization that in order to make highly active, specific, and well-behaved protein degraders from a heterobifunctional perspective, it requires a level of investment, commitment, and resources that was not anticipated initially. And
Mm.
so I think human nature tends to gravitate to the lowest activation barrier. And so the activation barrier for a molecular glue is apparently lower, but I actually believe that for actually making the final drug is technically higher because actually the rules are very different and the risks are generally higher. But we can talk. I love to talk about that.
So the field, when I think of like the literature on induced proximity, is like growing rapidly, right? And there are these other cellular systems, new ligases being proposed, trafficking pathways, other functions beyond degradation. As you kind of look to the future of this field, what do you think could offer a next chapter?
Yeah, I love actually that a field is being created by heterobifunctional molecules, right? As you said, I'm a chemist by training. So basically, we're enabling through understanding on how to make effective heterobifunctional bispecific molecules, new pharmacology. So degraders through the UPS system, I would call it, it's probably the most advanced of these technologies. I like thinking about extracellular uh, bispecific molecules to enable other type of protein clearance. I like the concept of sequestration of particular targets in particular cell type via bispecific heterobifunctional molecules. So I think, human imagination is amazing and the field is evolving. And our mandate at Kymera is that while we have, I think we have perfected as much as one can say on this earth, the heterobifunctional degraders probably as well as one could we have to continue to innovate ourselves. And so we are looking at other ways to use these highly sophisticated now chemistry to solve other type of problems.
Yeah, that's very good. Because I wanted to ask you, with such an expanding literature and so many directions that it's going, like given the stage that your company is at, right, with the level of platform, optimization you've done, like what would be your threshold for saying, okay, we're going to incorporate something new? Like this is more than incremental. We need it.
Yeah. That's a great question. So I always think about everything that we've done, and generally everything I've personally done is what problem we're trying to solve. And I always come down to the problem is what clinical problem we're trying to solve. So we will never go into a technology because it's cool or it's fascinating. I think we just don't have the time and money to make investments based on the technical attractiveness of a particular technology. So, there are lots of targets intracellularly that have yet to be fully drugged, for example, in immunology. So lots of work is going in there. And then actually staying in the same space, I think about there are targets that are outside of the cell, for example, that have yet to be drugged fully, or at least not conveniently, can we apply heterobifunctional chemistry to those type of targets through maybe different elimination mechanisms? So those are, again, I always start from target disease, patient population, opportunity to change the paradigm. So if we have those, we'll pursue those.
All right. Well, we need to take a little break, but stay tuned because when we get back, we will move from the sciencey discussion to Kymera's translational strategy. Back shortly.
The BioCentury Show is brought to you by the 12th China Healthcare Summit in Shanghai. From in-licensing to new codes, cross-border deals from Asia are transforming global biopharma. Meet top decision makers and investors at the 12th China Healthcare Summit to explore emerging opportunities and tackle key challenges in this rapidly evolving landscape. Join us in Shanghai, October 22nd to 24th to get a first-hand look at China's dynamic life sciences ecosystem. Register and learn more at biocenturychinasummit.com.
Hello, we are back with Nello Mainolfi, CEO of Kymera Therapeutics. In our first segment, we talked a little bit about the science of induced proximity, heterobifunctional molecules in particular, Kymera's specialty, but also where the field is going. And now we're going to talk a little bit about how you translate a new therapeutic modality. So when Kymera was starting out, the way I think of it is you faced a classical challenge that kind of every platform company faces, which is that, you had to choose a first target, a first molecule and indication, and you had to balance technology risk against biology risk. And, we often see companies when they're launching new modality platforms begin with a target that already has drugs against it. So it's well validated, well understood. That lowers biology risk, but then it raises the bar for differentiation, right? So there's just a lot of factors to balance. So how did you think about those trade-offs in your first program?
Yeah, no, great question. And I go back, I remember so clearly about, again, 9.5 years ago when we were having these conversations in the Atlas Venture offices about, how are we going to face this particular question. And we made, I think, both the bold and right decision to say we believe the concept of using small molecules to degrade proteins has been validated already. As you know, there are drugs that are approved that degrade proteins to the UPS. I would say the concept of using heterobifunctional molecules to degrade proteins therapeutically was the new angle. I'm a big believer, again, as a chemist by training, that you cannot change biology. Biology is almost impossible to change. Although now you can edit genes, etc., so you can do crazy things like those. But generally, you cannot change biology, but you can change chemistry. And if you have a chemistry problem, it's all about time and resources. You will solve the problem. So I took the bold approach of this is a validated technology, so we're going to go after targets that have not been drugged already. And so with that, obviously, came the risk of taking, let's so call it a technology risk and a biology risk. But how we decided to, I believe, dispense of the biology risk is to focus on molecular pathways. All the pathways that we pursued, we put 5 molecules in the clinic in the past, actually 5 years. All the pathways had upstream clinical validation where the upstream node on the pathway and be drugged successfully and safely. Most of our targets, I believe at least 80% of them, had human genetics data telling us that you can actually remove, there are people that walk around without that target and they have a normal life, and or that deletion of that target is beneficial in that particular disease context. So while that target hadn't been drugged, but the pathway had been drugged and there was strong human genetics data on that target. And to be honest, I look back, we have decided to prioritize some programs and deprioritize others, but I have no regrets about how we went about target selection. And I think, and hopefully you'll agree, and the listeners and viewers would, that we've actually inspired the field with our choice of targets.
Yeah, well, let's talk about your STAT6 program. So that's STAT6 is downstream of IL-4 and IL-13. So those are the targets of Dupixent, right? A very successful drug. So, it makes some sense that it's in that pathway that you could lower the biology risk. On the other hand, it's an intracellular target. It's involved in probably other things going on in the cell, meeting and other types of signaling, or at least it might be. So how important is it to you to build readouts into your early clinical studies that can more fully flesh out this specific mechanism, right? Versus say keeping those studies as lean and cost efficient as you can manage, because I feel like that's a balance companies also have to strike.
Yeah, no, that's a great question. I would say that, as I always tell my team, we have the sense of urgency and the focus of small biotech, but we have the rigor of larger pharmaceutical companies. And so when it's about doing the right thing, we take no shortcuts. So for STAT6, I could talk for days, so I'm going to try and be exceptionally efficient. What we knew already was that it's a unique transcription factor because from what we understand based on the biology, it is the selective transcription factor for IL-4 and 13. It actually is recruited to the IL-4 receptor alpha when IL-4 and 13 bind to it. So there's really nothing in between the receptor of STAT6. There's obviously a kinase that phosphorylates STAT6, but when it's bound to the receptor. So we knew that there was this one-to-one correlation. Then we knew obviously the pathways being exceptionally validated with Dupixent and other IL-13 biologics. So what we needed to de-risk is there any other role of STAT6, right? And so we looked at human genetics. There are humans that have gain of function of STAT6 that have severe allergic diseases. So it's telling you that STAT6 role is Th2 allergy. Also, there are humans that have heterozygous loss of function. They're absolutely normal. And so then we built a huge data set preclinically to ask the question, if you block STAT6, what is the downstream biology? If you block the IL-4 receptor, what is the downstream biology? In our hands, we always saw 100% overlap. Wherever we activated the receptor and we looked downstream, whether blocking the receptor, blocking STAT6, we saw the same biology. So when we went into the clinic in humans, the question is, do we see any other downstream effector that is modulated by STAT6 that wasn't seen with dupilumab? And again, I can only speak to the healthy volunteer data. Everything continued to track as if you block STAT6, you can have a dupilumab-like profile with an oral drug.
But how broadly are you doing mechanistic work in the humans in the clinic? Are you doing omics studying? Are you asking sort of open-ended questions, or are you keeping it very focused?
So I can say we've used human cells extensively preclinically, and we looked at transcriptomic profiles at all type of downstream signaling downstream of the receptor. And we've showed that our transcriptome changes overlaps for all intents and purposes to what dupilumab IL-4 receptor blockade is. In the patient study, which is a study that we just recently concluded dosing and we're sharing in 4Q actually in this quarter, we're actually looking downstream in the skin lesions of AD patients at the whole transcriptome of these patients. And we are asking the question, besides Th2, are we seeing any other changes that are not, let's say, predicted by a dupilumab-like response? So we're looking at that as well.
Very cool. Yeah. Well, let's talk about partnerships. Biotechs have to navigate this decision as well, like what to keep in-house, what to partner out, and when, when is the right timing. And so you have a few, Sanofi, Gilead. How did you make those decisions for Kymera?
Yeah, I think this is an industry that relies on efficiencies and on kind of working with the other, let's say, stakeholder to deliver medicines to patients. And so we cannot build these companies with the approach of everything needs to be done internally. Both biotech companies and also pharma companies. I think there is that continuum of innovation that relies on these kind of unspoken rules of creating efficiencies. So I think there are partnerships that make sense depending on the stage of the company. We've actually done 4 partnerships. We had an early one with GSK. We did a platform partnership with Vertex, an asset-centric partnership with Sanofi, and one with Gilead. We're at the stage now where we're well capitalized. We have all the plans that we have for a wholly owned pipeline are funded through the second half of 2028. And so for us, partnering is about potentially expanding the patient impact. So for the bar is extremely high for us to partner programs that are central to the Kymera story. So in immunology, it's going to be extremely difficult. For sure, we're not partnering before the end of proof of concept phase 2b studies. And then the question after that, especially if we're successful across multiple programs, it will become around, can we do justice to these drugs? And as a growing company, execute on all the opportunities, or in some cases, does it make sense to partner for the greater good? And I think this is a decision that is not in the immediate future.
Yeah, yeah. Well, so Kymera has been, you're like 9 years old now, and you're well capitalized, and you've got some clinical data that's positive, so congratulations. But I was thinking, like, for the sake of other companies, if you were founding, say, Kymera today in the funding and climate that we're all experiencing, do you think, as a platform company, you'd have to approach the business model any differently? Do you have any advice there?
Well, I would say that when we founded Kymera in 2016, it wasn't the hottest biotech market. Obviously, when you're getting a company off the ground, you're basically trying to build data to survive. And so it's a bit different than building pipeline optionality and ownership, etc. But the way I think the rigor that we've always used at Kymera is you have to earn the right to grow. Money does not give you the right to grow. You don't hire people or build programs because you have money. You do it because you show that your early data is giving you confidence that your innovation engine is reproducible and you have the team in place to execute. So we've always been very, very focused on not overextending ourselves. The second is you have to do the right thing, meaning there are no shortcuts to making drugs. And I think it's important that you run the right study for the eventuality of you needing to go all the way, because again, there is no shortcut to positive clinical data, which requires a level of rigor and commitment that sometimes we forget, let's put it this way. And third, I think you need to marry the technology to the problem. I feel the field of protein degradation has suffered from let's degrade the protein because we can versus let's degrade the protein because we want to. And I think that applies to any technology.
I think that is so true. Do you think the things you need to show really quickly to de-risk are any different in today's climate, or do you think it's the same?
Well, so we've been public for 5 years. So we've been dealing with different challenges, let's say, than an early stage biotech that is trying to raise private capital.
That's fair.
But I will say that in my experience, investors are looking for ability to trust a team that is being transparent about the great things that they have, but also the challenges they're facing. They want to see data that speaks to, especially if you're working on, let's say, a platform-based company, that data that speaks to the reproducibility of the platform and not necessarily only on the fact that one program is going to work, at least the type of investors that I like to spend time with. And I think, the importance of understanding that making drugs take time. And I've been at fault myself when we come up with these timelines in early stage of the company. We tend to be over optimistic, and that creates actually inefficiencies and misalignments with people on the other side.
Yeah. Okay. Well, we're just about out of time here, so maybe I'll just ask you a closing question, which is, if we were to talk again in 4 or 5 years, what would you hope people will be saying then about Kymera's contribution to this field?
I think that we transformed immunology with our degraders. That's what I hope people would be able to say, because that's the goal.
Very good. Yeah, I feel like new modalities so often start in cancer. So it's kind of neat to see you jumping into immunology sort of straight away. But thank you so much for your time. This has been a very interesting discussion. And I hope to talk to you again soon.
Thank you. Likewise.
Thanks.
Brought to you by the 12th China Healthcare Summit, returning to Shanghai October 22nd to 24th. Meet top decision makers and investors and get a first-hand look at China's dynamic life sciences ecosystem. Learn more at BioCenturyChinaSummit.com.
Kendall Square Orchestra provides the music for the BioCentury Show. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.